if you want to understand how a massive scientific breakthrough actually happens, don't look for a lone genius in a falling apple.
Right.
Look for a top secret 2 million page bureaucratic archive and the artificial intelligence required to read it.
Yeah, it's so true. I mean, we love that romantic myth of the sudden epiphany, right? Like Alexander Fleming just staring at a moldy Petri dish and suddenly discovering penicillin.
The classic light bulb moment.
Exactly. But modern biology operates on a completely different paradigm. It relies on this massive, sprawling, and honestly largely invisible infrastructure.
Welcome to the deep dive. Today, we're taking you into the hidden plumbing of the genomics revolution. We have this absolutely fascinating 2026 paper from Nature Communications.
It's a great paper.
Yeah, and the researchers essentially built an AI time machine to read decades of internal government memos. They wanted to figure out how a massive state-funded agency actually orchestrates science behind closed doors.
Right, because that's where the real groundwork happens.
Exactly. But before we pour these millions of pages into the AI, let's make sure we're grounded here.
Yeah.
What exactly is the difference between genetics and genomics? Because people mix those up all the time.
Yeah, it's a really crucial distinction. Think of traditional genetics as studying a single tree in a massive forest.
Okay, just one tree.
Right. You're looking at one specific gene, maybe trying to find the exact mutation that causes something like cystic fibrosis. Genomics, on the other hand, is mapping the entire forest ecosystem simultaneously.
So you're zooming way out.
Exactly. You're looking at large sets of genes, or even the entirety of an organism's DNA, and how all those parts interact with each other. And today, genomics accounts for at least 20% of all biomedical research into human genes.
Wow, 20%.
Yeah, it is an absolute juggernaut. And the catalyst that started laying the pipes for this entire field was the Human Genome Project that was run by the National Human Genome Research Institute, the NHGRI, along with the Department of Energy.
And the NHGRI, they kept all the receipts.
Oh, they definitely did.
They have this meticulously curated digital archive called the Core Collection. It basically covers the Human Genome Project and everything that followed it. Initiatives with acronyms like ENCODE, HapMap, and LSAX.
Right.
We're talking about 22,843 individual documents. That is over 92,000 pages, mostly spanning from 1993 to 2007.
And it grows by about 5% every single year, so it's huge. But the problem for historians is that you can't just hand decades of raw, unredacted government memos over to an open source AI to summarize.
Right, because of privacy.
Exactly. These documents are absolute landmines of personally identifiable information. I'm talking social security numbers, private emails, really candid performance reviews.
Yeah, you don't want an AI leaking somebody's bad performance review from 1998.
No, you really don't.
So how do you safely analyze that kind of volume? I mean, if a human tried to read and redact 92,000 pages, they'd be working for decades.
Well, you build a computationally augmented mixed methods pipeline.
Say that 3 times fast.
Right, but basically, think of it as a highly advanced, multi-stage digital processing plant. Step 1 is page stream segmentation.
Okay, what is that?
So when old archives are digitized, librarians often just feed thousands of loose physical pages into a high-speed scanner, which just spits out one gigantic PDF file.
I hate those. Just one endless scroll.
Exactly. So the AI has to somehow figure out where one memo ends and a completely unrelated grant proposal begins.
But how does it do that without a human checking every page?
It uses visual and structural algorithms. The model looks for layout cues, so maybe the sudden appearance of a formal letterhead or a shift in the margin structure or, a signature block at the bottom of a page.
Oh, I see. It uses those spatial clues to physically slice that massive PDF back into individual coherent documents.
Yeah.
Like chopping a giant block of ice into individual cubes.
That's a perfect analogy, yeah.
Okay, so what happens after you've sliced it up?
Step 2 is where they deploy computer vision for handwriting removal. And this is a fascinating mechanism. They use an algorithm that actually maps the contours of the ink on the page.
Wait, hold on. If you remove the handwriting, aren't you deleting historical information? I feel like the handwritten margin notes from some director are where all the juicy historical gossip actually lives.
You're absolutely right, and it is a massive trade-off that the researchers had to make. But here is the mechanical reality optical character recognition, or OCR, which converts images into searchable digital text. It relies on bounding boxes.
Yeah.
It's trained on predictable, straight lines of standard printed fonts. It fails miserably at the chaotic, unstandardized scrawl of human handwriting.
Oh, right. It just outputs a bunch of random symbols and wingdings.
Exactly. But more importantly, handwriting is overwhelmingly where the most sensitive data lives. It's the quickly jotted down personal phone number or an off-the-cuff opinion about a colleague's competency or even a doctor's note.
So it's basically a liability.
Exactly. So the computer vision model looks for the lack of a straight baseline, the erratic variability of stroke width, and it physically erases those pixels from the document before the text is even extracted.
Wow.
Yeah, it sacrifices a bit of historical context to ensure a watertight seal on privacy.
A necessary sacrifice, I guess, to make the whole project legally and ethically possible.
Yeah.
So now we have clean, typed, extracted text. What's the final step?
Then comes the final filter, Roberta.
Roberta.
Yeah, it's a really powerful natural language processing model trained specifically for entity recognition because unlike old software that just looked for any 9-digit number and assumed, hey, that's a social security number.
Right, which gives you tons of false positives.
Exactly. Roberta is bidirectional. It reads the context of the words before and after the number to understand its semantic meaning, so it can tell the difference between call me at and his ideas.
Well, that's smart.
Yeah. It intelligently masks out names, addresses, and financials, leaving behind a completely anonymized data set.
So just to recap this for you listening, these multi-layered AI pipelines allow researchers to safely digitize and analyze massive historical archives by automatically filtering out private data. They're basically building a highly advanced digital sieve.
A digital sieve, exactly. It perfectly catches sensitive personal data while letting the golden nuggets of historical scientific data flow right through.
It really makes you wonder, if you had this kind of AI sieve running on your laptop right now, what hidden historical archive would you want it to safely read and summarize for you?
Oh, man. It opens up a whole new world of historical analysis because once researchers finally poured those 2 million pages through the sieve, what came out the other side completely rewrote the timeline of modern biology's most powerful tool.
Right, and this is the part that genuinely surprised me. Let's talk about GIS.
Genome-wide association studies. This technique is an absolute powerhouse today.
It really is. And to understand why it's a powerhouse, we need to look at how the mechanism actually works.
Okay, lay it on us. Imagine you want to find the genetic cause of a specific type of heart disease. You take a massive population of people who have the disease and a massive control group of people who don't. GWAS rapidly scans across the entire genomes of all these individuals.
And a genome is what, roughly 3 billion base pairs long?
Yes.
So it's looking at 3 billion data points per person.
Right. And it's looking for single nucleotide polymorphisms, or SNPs.
SNP.
Yeah. SNP is simply a single letter variation in the DNA sequence. Imagine having 2 identical billion-word libraries, but in one library, a single word is misspelled.
Okay, I can picture that.
GWS does the staggering statistical math to find exactly which tiny genetic typos show up consistently in the disease group, but not in the healthy group.
That's incredible. And the impact of that math is undeniable. The paper notes that GOA studies are in the top 0.6% of all scientific techniques for citations.
Oh, yeah. They have flooded the scientific literature with previously uncharacterized genes, linking them to specific diseases. It is the absolute bedrock of modern precision medicine.
But here is the historical reveal. If you look at the public record, the very first successful GRS paper was published in 2005.
Right.
But the AI found something completely different in the archive. It found internal NHGRI documents actively discussing GRAs way back in March of 1997.
Yep, 1997.
That is an 8-year gap between the government talking about it behind closed doors and the scientific community actually using it. Why the delay?
Well, it comes down to the plumbing. In 1996, a paper in Science proved that the math behind GRA was theoretically possible, but you couldn't actually do it because scientists lacked a map.
A map of the typos.
Exactly. You can't search for tiny genetic typos across 3 billion base pairs if you don't even have a baseline map of where those common variations usually occur in the human population. The NHGRI leadership realized this in 1997.
So they didn't just wait for some university lab to figure it out.
No, they saw the bottleneck. The archive shows they actively convened external experts and then intentionally designed and funded the International HapMap project starting 2002.
And what was that?
The entire purpose of that multi-million-dollar project was to build that exact SNP map.
I have to pause here because this completely shatters how I previously thought about science funding.
Right.
I always pictured agencies like the NHGRI as giant passive administrative inboxes.
Right, like a suggestion box.
Yeah. A brilliant scientist has a great idea. They submit a grant proposal. The agency reviews it and says, sure, here's some cash. But this shows they weren't passive at all. They were literally engineering the playing field almost a decade in advance.
They were laying the massive, expensive pipes that no single laboratory could ever afford to build on their own. They knew that once they turned the water on, once they released the HAP map, the entire scientific community would be able to run with it.
And that's exactly what happened in 2005.
Precisely.
So just a quick recap here. Transformative techniques like genomics don't just magically appear. They require years of hidden, coordinated investment in foundational infrastructure by funding agencies.
Exactly. It's a long game.
It really is.
Yeah.
And looking at the science we see in the news today, it makes you ask yourself, what invisible infrastructure are these agencies quietly building right now that's going to define the breakthroughs of, say, the year 2035?
It's a great question, but that proactive approach raises a really difficult logistical issue. If an agency is building the infrastructure, they have to decide exactly where to lay the tracks. Resources are finite. How do they actually pick the winners?
Right, which takes us into the sequencing lottery. So the deep dive zeroes in on a specific committee within the NHGRI called LSC. That stands for Large-Scale Genome Sequencing and Analysis Centers.
Right. And back in the early 2000s, sequencing a whole genome was still outrageously expensive. So LSC had to decide which non-human organisms were going to get sequenced next.
You can imagine the lobbying that went on for that. Every biological subfield wanted their organism sequenced. Do we map the cow, the flatworm, the sea urchin?
Oh, the pressure was immense because if your community's organism gets picked, your entire field gets modernized overnight.
I'm just picturing the cutthroat backroom dealings of the global sea urchin lobby.
It was probably pretty intense.
But to figure out how the committee actually made these subjective choices, the researchers didn't just read the memos. They used a machine learning tool called CatBoost to mathematically reverse engineer the committee's brains.
Yeah, CatBoost is a really powerful algorithm. It uses something called gradient boosting on decision trees. Basically, it's excellent at finding patterns in messy categorical data.
Okay.
So the researchers fed the algorithm 4 distinct categories of features extracted from the historical proposals to see what actually predicted a winner.
Let's break those down. What was the first one?
First were the biological features. This is straightforward, things like the physical size of the genome being proposed, because a larger genome simply costs more money to sequence.
Okay, the biology is the baseline. What's next?
Second were linguistic features. And this is where it gets really fascinating. The AI analyzed the text of the proposals themselves.
Like how well they were written.
Essentially, yeah. It measured lexical diversity. Did the authors use a rich, varied vocabulary, or did they just repeat the same buzzwords over and over? It also measured how argumentative or forceful the writing was.
Wait, how does an algorithm actually quantify if someone is being argumentative? Does it just count exclamation points?
No, it's smarter than that. It looks at the structural complexity of the sentence trees and the density of absolute modifiers.
What does that mean in practice?
So if a proposal says, this sequence will prove our hypothesis and must be prioritized, that scores much higher for argumentativeness than a proposal that says, this sequence might suggest new avenues of research.
Oh, I see. It's quantifying confidence and persuasion. It's analyzing the sales pitch.
Precisely. Now, the third category was organizational features. For instance, did the proposal cleverly bundle multiple related organisms together to demonstrate, like, economies of scale?
Smart. Buy one genome, get a related species half off.
Exactly. And finally, the fourth category was reputational features. Specifically, the algorithm looked at the H-index of the lead scientists submitting the proposal.
Let's quickly define the H-index for a second, because it is the ultimate academic flex.
It really is.
It's a metric that balances how many papers you've published with how many times those papers have been cited by other scientists. So if you have an H-index of 20, it means you have published at least 20 papers that have each been cited 20 or more times.
Right. It's a rapid shorthand for historical track record and influence. So CatBoost takes the biology, the linguistics, the organizational bundling, and the reputation, and it tries to predict which proposals LSCC actually approved.
And here's the stat that blew my mind. The machine learning model predicted the human committee's actual historical decisions with an AOOC of 0.94.
Yeah, that is staggeringly high.
Now, I have to push back here. Predicting human committee behavior with 94% accuracy seems suspiciously high. Human beings are notoriously fickle, you know? Was the committee just rubber stamping a strict rubric, or was the AI finding something else?
That's a great instinct. But first, let's clarify OROC area under the receiver operating characteristic.
Okay, what does that mean in plain English?
It's not just a flat pass-fail accuracy grade. It measures the model's ability to correctly rank a random winner higher than a random loser across all possible thresholds.
Got it.
O1.0 is absolute perfection. So a 0.94 means the model achieved incredible separability. What it implies is that while human committees might feel subjective or fickle to us, they're actually heavily pattern-driven.
So the AI proved that it wasn't just about having the best science. It's almost like a college admissions matrix.
Exactly.
You can't just have perfect grades, which is the biology part. To actually get in, you also need a highly persuasive, confident essay, the linguistics, and a proven resume of past success.
That's the perfect analogy. No single feature could predict a winner on its own. It was the specific combination of all four that tipped the scales.
So to summarize this section for you, funding decisions are highly complex matrixed choices. But the fact that human subjectivity can be so accurately modeled by AI really poses a wild hypothetical.
Oh, for sure.
Like if an AI can reverse engineer a grant committee's choices with a 0.94 AUROC, do you think an AI could or should ever replace the human committee entirely?
It's a huge debate, and science administration is going to have to face it very soon. But, all of this plumbing, all of this matrix decision-making, it leads us to one ultimate question. We know how the winners of the sequencing lottery were picked, but did winning actually matter?
Did the water actually flow differently because of the pipes they laid?
Exactly. And to prove that structurally, you need a control group, a counterfactual.
Like in a clinical trial.
Right. In a trial, you have a treatment group that gets the drug and a placebo group that doesn't. If you want to prove that the NHGRI providing a sequenced genome actually drove scientific innovation, you can't just look at the winners.
You have to look at the losers.
Yes. The organisms that were formally proposed for sequencing, evaluated by the committee, but ultimately rejected.
Okay, so how did they do that?
Well, the researchers used the archive to build a pristine control group of those rejected organisms. Then, they scanned the published biomedical literature that came out in the 5 years following the committee's decision.
Oh, to see what the scientists were up to.
Right. They tracked specific keywords and methodologies to measure how quickly the scientists studying those organisms adopted modern genomic approaches.
And the data was completely undeniable.
It really was. In the 5 years following the decision, research on the approved organisms saw a significantly higher, mathematically distinct rate of adoption of genomic methods compared to the rejected group.
So, providing a foundational resource like a sequenced genome isn't just a shiny trophy for a lab. It actively forces an entire scientific community to evolve.
Yes.
If you give the flatworm researchers a comprehensive genetic map, they stop doing science the old, slow way. They immediately pivot and start using advanced, high-throughput techniques. The infrastructure literally dictates their behavior.
It mathematically accelerates adoption. This entire deep dive proves that scientific innovation requires immense programmatic effort.
And you mentioned earlier that it surfaces the humans who manage that effort too.
Yes, absolutely. Throughout the documents, you repeatedly see the name Dr. Elke Jordan. She was an unsung deputy director at NHGRI. And while the celebrity scientists were getting the magazine covers, she was quietly providing the vital, steady continuity between these massive decadal projects.
She was the architect of the invisible plumbing.
Yes.
And that is exactly what this all boils down to. When we realize that foundational resources act as an undeniable catalyst for human behavior, it makes you look at other fields of society. What other areas, maybe renewable energy or space exploration, desperately need a foundational map right now to trigger their own revolution?
It completely reframes how we view progress.
We spend so much time obsessing over the lone tech billionaire or the celebrity founder who claims they're going to single-handedly invent the future in their garage. But this archive proves that the true engine of innovation is massive data sharing, hidden infrastructure, and quiet administrators coordinating efforts over decades.
It's a team effort. A really, really big team effort.
So as you go about your day, ask yourself, who are the quiet administrators building the invisible foundations for the year 2050 right now? Thank you for joining us on this deep dive. Keep questioning the structures behind the science you read about every day. And remember, sometimes the most important discoveries aren't the apples falling from the tree, but the hidden roots that grew the tree in the first place. Catch you next time.